The organism I selected for this project is Borrelia burgdorferi, most commonly known for its ability to cause Lyme disease in humans. This bacterium is a flexible helical spirochete that is pleomorphic or able to change morphology based on environmental conditions. A unique characteristic is that its flagella is actually located between the inner and outer membranes in the periplasm. This allows the bacterium to better move through viscous layers, which, to no surprise, aids in its ability to invade a host tissue. Another characteristic is the outer membrane surface proteins, or OSPs. OSPA and OSPC likely play a role in transmission of the disease. Inside the gut, OSPA proteins are more present, indicating they help keep the bacteria in the tick gut. However, inside an infected mammal, OSPC proteins are more present, indicating that they may play a role in releasing the microbe to the tick salivary gland so it can release into the host during attachment. B. burgdorferi contains around 910 kilobase pairs in its linear chromosome, as well as 610 kilobases of plasmid DNA, both circular and linear. Its phylogenetic classification is as follows: domain, bacteria; phylum, Spirocheta; class, Spirochetia; order, Spirochetales; family, Borrelia genus, Borrelia; species, burgdorferi. Two of its relatives are B. afzeli and B. guerini, which also can cause Lyme disease. Like many bacteria, it uses glucose and other carbohydrates for its nutrition. N-acetylglucosamine is the essential nutrient for its growth, which happens to be a component of chitin, which makes up the tick cuticle. The organism's genome also codes for several transport proteins like ABC transporters and enzymes, which likely allow it to bring in nutrients from the environment. Finally, its main method of ATP production is via substrate-level phosphorylation and the pentose phosphate pathway. B. burgdorferi likely does not contain the mechanics necessary to interact with other microorganisms. There are two pieces of evidence to support this. First, when there are increases in Pseudomonas, Bacillus, and other bacteria in the environment, B. burgdorferi numbers go down. In addition, it appears that there is not a huge diversity in the microbiome of tick's gut, which would allow B. burgdorferi to populate there. B. burgdorferi's interactions with other organisms play a key role in its ability to survive and its pathological impact. So the artwork I did here was to demonstrate the infection cycle of ticks. We start here with number 1, where an adult tick lays eggs. 2, those eggs hatch into larvae. Then, in number 3, the larva may feed on an infected mammal, thereby acquiring the bacterium. Here, the tick will grow into an adult, and then will feed on another mammal. And then later on, the tick will drop off this animal to lay its eggs, and the cycle continues. Now, at number 4, where there's an infected nymph, if that nymph happens to come into contact with a human and is able to latch on for long enough to pass B. burgdorferi, then that human may acquire Lyme disease. Upon a bite from an infected tick, there are 3 stages of infection. First, in days to weeks following exposure, erythema migrans may appear at the bite site. During the second stage, once the disease has spread via the blood and lymphatic system, people may experience headaches, neck stiffness, fever, or chills. They may also undergo neurologic abnormalities and, on occasion, cardiac problems. Finally, the third stage is marked by sporadic episodes of arthritis affecting large joints and/or chronic neurologic sequelae. Thankfully, there are many antibiotic options available for each stage of the disease. However, some people report ongoing arthritic and neurologic conditions even after excessive antibiotic treatment.